Hello everyone and welcome to the third and final talk of today's online Pint of Science Festival. I'd like to thank the University of Oxford and the University of Warwick for the wonderful talks today. And now I'd like to introduce you and welcome you to all to the University of Bath here in the southwest of England. I'm your host Jazz, and what a truly exciting lineup we have for you tonight. Today we'll be discussing the themes virtual pigs and hot wiring genes. To start, our first speaker, a tech wizard from the Department of Computer Science, will take us on an extraordinary virtual journey through optical motion capture technology using virtual pig avatars. And in hot wiring genes, our final speaker, a talented young PhD student from the Department of Biology and Biochemistry, will take us into the microscopic world to better understand bacterial evolution using genetic tools. And finally, we'll end with some question and answers with you, our wonderful audience. But before that, welcome once again to our show from wherever you may be in the UK or where else in the world. I thought I'd like to get started with a quick question for you all. I'm a scientist myself, and over the last 10 years, there have been a myriad of scientific advancements which have helped advance humankind, whether it be related in fields of computer science or medical technology. What I want to know is, from you, our lovely audience, in the last 10 years, what scientific information have you come across, whether it be on the news or the internet, that you found particularly interesting? I thought I'd give a quick example. And it's this little thing. What looks like a power adapter for a phone or a little USB device is actually a mini portable DNA sequencer used to decipher the genetic code. Because it's small and portable, this was used to help monitor the Ebola outbreak in West Africa, and currently 200 of these are being sent to China right now to help monitor the coronavirus pandemic. That's my example of an interesting advancement in science. But for now, it's that time of the night where we grab our favorite beverage. I'm here enjoying a cup of coffee, and sit back, relax, and dive into the science. Please do stay in touch with us throughout the entire show using the Twitter @ or the hashtag, or using the comment section below this video. I thought I'd like to introduce our first speaker. He hails from the Department of Computer Science at the University of Bath and is the head of studio at the Center of Analysis of Motion, Entertainment, Research and Applications. Some of his work is also featured on the big screen in some of the most recent Harry Potter films. Tonight, he's taking us on a virtual tour using virtual pig avatars. So without further ado, please raise a glass and welcome to your screens our first speaker, Mr. Martin Parsons.
How do you do? I'm now bringing my experience in visual effects as a visual effects artist to help researchers at camera to build quite often things like data sets. You might have heard people are using a lot of data sets nowadays for doing machine learning, so they can, which does require quite a lot of data. And we've got our own motion capture studio here. We've got numerous cameras we've got 10 sort of 4K video motion capture cameras for doing kind of volumetric capture, but we also got a 20 camera optical motion capture setup, and we've got various display devices for VR, AR, mixed reality and all that kind of thing, and we're using game engines for displaying, but we're looking into sort of the future of entertainment and also using the same kind of technology for health and sports and stuff like that. But we've found ourselves looking quite a lot into investigating quadruped motion capture, because this is quite an overlooked field. We've started with my dog, actually, Maggie, because she's actually quite amenable, and it's a similar problem for all sorts of quadruped motion. But as you can see, we have found why quadruped motion has been overlooked, because as you can see from this, working with animals does have its own issues in that they're not always as cooperative as you might like. Even a pet like Maggie, who is tenderly reasonably biddable. But one researcher was looking into the problem of working with motion capture, turning motion capture of 2D bipeds into quadruped motion, because it was working with a film production company whose idea was to take actors and actually but get them to behave like normal actors, but actually appear like animals. And as you can see from this, it's the best one in the world. Though they valiantly did their best to be moved like a pig's, you can see it's not really going to happen, even with the most sophisticated retargeting things that you have in motion capture. So what we had to do is then build a data set of pig motion, which then the machine learning could use to actually source and take the bipedal motion from the actors and then make it turn it into the appropriate pig motion. Now, of course, for this we needed a pig, so we went to our local farm, where I hadn't really realised actually quite how large farm pigs are. As you can see, this is with his prize boar, our local farm with his prize boar. And you can see we can't really get that pig into our studio. I don't think it would fit in the lift for a start. So the only thing we could do is actually take all our equipment down to the studio and set it in that yard behind. That kind of slightly shiny effect on the floor is actually pig excrement, which gives its own problems, really. But so fortunately, the farmer introduced to Molly. Molly's not quite as big as the boar. She's a female pig, as you can see. For this, we had to put on motion capture markers. You can see arrayed along the barriers along the top. You might be able to just make out the motion capture cameras. They look like polos on ends. And on Molly, you can see she has these markers. These are actually made of polystyrene balls covered in reflective tape. Because with optical motion capture, you just see, this is all that it sees. But so all these markers have to be put in quite crucial positions. And you can see from this, you can your brain can kind of make out a pig just about, but it's not really enough to do a 3D reconstruction. So if you have multiple cameras and they've all seeing these markers from the same direction, you can then make a projection from the center of the camera through the marker to make a 3D point. And then if you do that for all the points, obviously you get through all the markers on the pig, and then you're now getting the full motion of the pig. But again, this is not really useful in itself. In fact, this image, it looks pretty much the same. It's still like a 2D pig, but you can still see it. So now what you now need to be able to do is actually do something useful with those markers, because at the moment, none of those markers are actually identified. The system knows that there are markers in 3D space, but it doesn't actually know what they are. It doesn't know whether, what they're connected to. So what you have to do, first of all, is identify what all the markers are and how they relate to each other. And once you've done that, you can do something fairly interesting with that. You can then use that information to drive a skeleton. And this is all the markers using constraints to drive the skeleton. And once you've got a skeleton, you can then use that to drive a model. And there it is. And then once you've got a model, you can apply texture on it. And there you have your pig motion. Now, to get that model, we did attempt to scan it. But the problem is with the pig, as you'll see, it doesn't really do what you want. So we put the food down, but it kept moving. So you get a bit of the rump, and then you get a bit of the side, and then you really couldn't find it to get it to work together. So what we then decided to do is then maybe we'll get something that is static. So this is our toy pig. And because it doesn't move, but it's still a pig, even though it's this one's only a few inches long. Once you scale it up, you can put it on to the same, give it the same motion as the pigs, and gives it quite a good facsimile of a pig. And then when you've got them all together, you can make a pig party. So this is taking all the pig motion, all the different pig motions together, and then, stitching them all together and putting them all into the same scene. And what we did with this, even though the film never got made, we actually then decided to take all this pig motion and make them into virtual pigs. And we use this on a virtual reality. So then we use a virtual reality device. With this, if you put this headset on, I mean, it works much the same as other headsets, but rather than just having just a motion sensor and a direction sensor, HoloLens actually has scanning equipment on the front. It has this kind of structured light scanning equipment, and actually makes a rough scan of the whole room. And what it means, it can then place the pigs in the scene. So even though here we can see that there's a background, with an augmented reality device, you can just have the pigs appear, and because the headset knows where it is, as it turns, the pigs will be rendered in real time and will actually appear to be running around in the scene. I mean, the device is slightly limited in that the screens are fairly narrow. So, but I think the newer one is a bit better than that. And then there's an audio device here which will give you the pig sounds. But and what we have subsequently done is we've now taken the pigs and put them and rendered them in 360 video. So if you're interested in being able to see the pigs yourself, you can find our link on our blog post on our website, and here you'll find a 360 video of the pigs. So what's been happened here is taking all the pig motion, stitched them together in an animation software, and then rendered it as a 4K 360 video. And what you can do is you can take your phone or a headset, and as it looks around, it will use your positioning and orientation device of your phone, and it'll be able to view all around inside the 360 video. I'll see if I can actually find a version of that. I think I should have it. Just to give you an idea of what it's like. Let me just drag it over here. So there you can see with 360 video, you can actually, I mean, if you imagine you've got your phone on, you can then, if you hold it around, you pan around you, and you can actually see these pigs moving. What was quite nice, we did actually put some markers on the ears just to sells it quite well, really. Unfortunately, we couldn't really get any markers on the mouths because they kept knocking them off. Now it all sounds fairly straightforward, but actual fact, when you're actually doing all this in practice, it actually is a bit of a nightmare, because you get there. We went to the farm, and we said beforehand, we said, have you got any power? And he said, yeah, that's fine, I've got power. And we turned up with all our equipment, and what it was is one lead hanging down from the barnyard roof, which was somewhat alarming. So unfortunately, we couldn't have any heaters, and it was in the middle of December on a very cold evening, and we had to film at night because the infrared from the sun would interfere with the motion capture cameras. And then we had to, the farmer said that he wouldn't be able to, he would be able to get the pig. We said, how are we going to get the pig to do what we want? He says, well, I'll tell you what, I won't feed the pig for all day. But the trouble with that was, I said, well, how are we going to keep it still? He says, well, so then he threw a load of food on the ground. And then, of course, then I was able to put the markers on with my trusty glue gun. But unfortunately, then of course, then once it had all the food there, he couldn't. He didn't want to do anything else after that. So then the farmer then had to come out and get a great big board and move the pig around, or use a broom. But eventually, and all the cameras weren't in focus, and it actually was proved quite difficult, because although you can see that I'll just go back to the. Although you can see the markers there is something else that's you have to actually, in order to identify those markers, you have to have some kind of template within the system which actually recognizes where those markers are. So it has a kind of it's a template of the pig, and it tells you where exactly those are markers in relation to each other. Unfortunately, if some of the markers then get occluded, it'll swap with the markers of something else. That's when you get that kind of characteristic glitchiness in motion capture. And then if they don't, so then you quite often have to manually go through and then label all the markers again. Just to make sure they all work, and then you have to make sure that your motion is perfectly smooth, and that the joints are actually related properly and they're not popping everywhere. Because the problem is, you have to calibrate the system, and so that's what we got to do to get it to kind of move their joints as much as possible. So we got it to climb over a block of wood, and of course, as soon as it hit the block of wood, one of the markers fell off the hooves, so you lost all that motion. So you then had to fill that in again afterwards. So actually, though it all seems quite straightforward, actually in practice it's quite difficult. But now we have some lovely pig motion, and so if you want to see it yourself, I'll take you back to that link again. There we are. If you follow the link on the bottom, you'll see where you can have a view of the virtual pigs yourself and see what you think. And back to you, Jazz.
Thank you very much, Martin. That was a wonderful talk. We've got plenty of questions coming through, so you'll be busy after with the questions round. I'd like to say hi to Gil Harris. I was mentioning her interests in DNA sequencing. We've got Sue Chant as well, real science fiction geek, asking about the robotic exploration of Mars. I mean, that's a fantastic leap. Like recently, we've had SpaceX with their rocket going to the International Space Station. So the sky is literally the limit. Looking at Mars and potentially beyond, there's the whole universe out there to explore over the next 100, maybe even thousands of years. And it's amazing to see how quickly we've progressed from the initial Apollo 11 and the moon landings, the initial development of the space program. It's incredible. Once again, I'd like to thank Martin for his wonderful talk, and would like to again encourage the audience to ask questions. Our next guest is a young PhD researcher within the Taylor Group at the University of Bath, Milner Center for Evolution. His work focuses on creating evolutionary challenges for bacteria by removing genes and understanding how evolution can make up for these missing genes, thus giving us an insight as to why evolution might take one route over another. More recently, he's presented some of his work as an invited speaker for the Society of Molecular Biology Evolution, and in his spare time enjoys amateur wildlife photography and reading history. So without further ado, let's once again raise a glass for our next speaker, Mr. Matt Shepard.
Hi there, Jazz. Hi there, hello. Thank you very much for the introduction. So, as Jazz said, I'm going to give a talk about my PhD research, which focuses on using genetic engineering to try and better understand the natural process of evolution. So what comes to mind when you first think about the evidence for evolution? I think for a lot of people, it's fossils. So these are snapshots of an organism's evolutionary history and what that organism used to be like a very long time ago. This example is Archaeopteryx, which shows the sort of missing link between reptiles and birds and shows that those two groups are related to one another. But there's also lots of other evidence for evolution. A big one is comparative anatomy. So this is looking at the structures and biology of living organisms that exist today and comparing them to see for any similarities and relatedness. So these are 4 different limb structures in 4 different animals, which all have different functions that they've evolved to do. But they all have the same bone structure, as you can see from these different colors, whether it's a wing or a flipper or a hand. So this shows that these structures and animals share a common ancestor, that they're related to one another because of these shared structures in their limbs. We can also study evolution using molecular evidence, and this has been a really big thing with the advent of DNA sequencing. So we can take the DNA and work out the sequence of letters which encode information and then compare that between organisms to create a tree of life. And DNA sequencing has been very revolutionary for the understanding of how organisms are related to one another evolutionarily. For example, it was long thought that plants and fungi were closely related to one another because they both grew in the soil and were not mobile, they can't move around like animals. But DNA sequencing highlighted that actually fungi and animals are quite closely related to one another rather than closely related to plants. Can we observe evolution in real time? This is a big question because all of this evidence that I've highlighted already is essentially historical or comparing living species to infer evolutionary relatedness. So can we actually observe the process of evolution happening? Now you might think, hang on, evolution takes far too long to occur. It's millions of years, right? Well, actually, the rate of an organism's evolution depends on its generation time. So essentially, how long does it take for an organism to reproduce itself? So for some organisms, this is simply far too long. So the sequoia tree, for example, takes 100 years to reproduce itself. And then humans and other primates, we're talking about generation times of decades. Because you need several hundred generations to observe any evolution happening, you would need centuries for the study of the evolution of these organisms in real time. Of course, scientists like to have their results in their own lifetime, so this isn't really a practical study system. But there are animals that, of course, reproduce faster, small mammals and rodents, for example. The house mouse can reproduce at a rate of about once every 2.5 months, so it can reproduce a lot quicker. And that means that you can observe its evolution happening, albeit still over a long period of time, several decades, for example. Going even smaller still, and some organisms reproduce incredibly quickly, about 20 days for fruit flies and 20 to 60 minutes for bacteria. This means that bacteria especially, and that's the organism that I work on, evolve very quickly, and you can study their evolution within the space of a few weeks or months. So this was noticed quite early on and led to the development of a field called experimental evolution. And this was the first evolution experiment to have occurred. It might have occurred a lot earlier than you might have thought. It was actually with a guy called William Dallinger in the 1880s. So this was a time when, to be an experimental scientist, you needed to have a lot of money and a lot of free time. So naturally, as an English priest, William Dallinger had plenty of both. And he used his spare time to set up a rather elaborate system of tanks containing broth. And in that broth, he put microscopic, what he called microscopic animals, or microbes. And he noticed something, that at 15 degrees Celsius temperature, the bacteria would grow in the broth, but at 70 degrees Celsius, they would no longer grow. But then what he did was quite interesting. He increased the temperature slowly between 15 and 70 degrees. And he did this over a period of 6 years. And what he found was that after that experiment, the bacteria or the microbes would then grow at 70 degrees and not grow at 15 degrees. So what he took this to mean was that the microbes had adapted to now live at the hotter conditions. So he saw it as evidence for evolution in action. There was a bit of a pause after his work, and then a lot of progress happened in the late 20th and early 21st centuries. A key milestone is the long-term evolution experiment. So this was set up by a guy called Richard Lenski in the 1970s in America, and along with various colleagues throughout the years, he has been evolving E. coli. You may have heard of E. coli. It's a bacterium that lives in your small intestine and is adapted to live in that environment. He took this bacterium and placed it into broths, again, a sort of nutrient soup, and every day after incubating those broths, transferred some of the bacteria to a fresh pot. This meant that the bacteria never ran out of food. And he's done this every day since 1978, and it's still going now. I heard it's been put on pause for the coronavirus pandemic, but they plan on restarting it after the pandemic has passed. But in those 32 years, the bacteria have adapted to live a lot better in the broth than they had previously. So they grow faster, they use nutrients more efficiently. They've even evolved to utilize nutrients that they couldn't eat before. So they've essentially changed their diets in response to the new environment that they found themselves in. And this has been going for so long that he's studied effectively a very long period of evolution. So it's about 78,000 bacterial generations. If you turn that into an equivalent in human generations, it's 1.5 million years of equivalent evolution. So it's a very long time frame evolutionarily for this microbe, and he's seen all manner of quite interesting changes. There's this field of experimental evolution, and this is what I'm doing my PhD in. Working on bacteria has a set of advantages. Not only do they reproduce really quickly, making the entire enterprise viable, you can also fit very large population sizes into a very small container. So you can fit millions of bacteria into a very small volume of liquid. You couldn't do that with mice or something. You'd need a huge warehouse to house that sort of numbers. So that's a big advantage because, of course, evolution happens to populations, and with a bigger population size, you have more chance for variety and mutation. So it's a lot better to use this for a model system. You can also cryogenically freeze bacteria. That effectively stops their evolution and keeps them in stasis. And then years or decades later, you can thaw them out and then study them as if nothing's changed. And that means that you can create a frozen fossil record of bacteria down the ages of your evolution experiment. And that means that you can directly compare the descendants of a bacterium with their ancestor. And that's something you can't really do with any other organism. As of yet, you can't cryogenically freeze a mouse or something and then revive it later. So this is really a fantastic benefit of working with bacteria. But we also have a molecular biology toolkit for studying evolution in real time and studying bacterial evolution. Of particular note is the cost of sequencing, of DNA sequencing, which has become incredibly cheap in the last 2 decades. To begin with, back in the early 2000s, sequencing the human genome, as shown on this graph, cost about $100 million. And now the cost of that is about $1,000 after 2 decades. So that's a huge drop in price. Bacterial genomes are a lot smaller than the human genome, so they're even cheaper. I can get a bacterial genome sequenced for about £70, which in terms of a research budget is really quite cheap. So you can get quite a lot of sequencing done. And sequencing is really important for the study of evolution, as it lets you identify mutations that have happened, and that's the raw material for natural selection. You also have a toolbox, a molecular biology toolbox. Bacteria are easy to genetically engineer, or relatively easy to genetically engineer. And that means that you can essentially make any change that you'd like. You can remove genes, add genes in, change the timing of when genes are switched on or off. And that can allow you to create interesting situations to study bacterial evolution in. So that's what I do during my PhD and my colleagues do. That's creating challenges for evolution by using genetic engineering in this sort of experimental setting. So my work focuses around the bacterial flagellum. It's these filaments you can see in the photo in the bottom left. It's essentially like a tail on the bacterium and is spun around a bit like a ship's propeller and allows the bacterium to swim along through a liquid. This propeller is hooked up to a control switch, and that control switch is called FleQ. Essentially, that switch determines whether the bacterium makes the flagellum propeller or not. And that's important because bacteria don't always want to make the flagellum. Sometimes they want to stay still and don't want to move around. And it would be wasteful to make a flagellum that they're not using. This control switch is hooked up to a control circuit. Think of it like an electrical circuit, which takes inputs from the environment. It'll sense situations such as there's food over there, or there's a threat coming to me from that direction, and will signal to the control switch to switch the propeller on or off. So what my supervisor did and what we work on, she removed the switch itself. So that's FleaQ, and actually did that using genetic engineering. So the gene for that switch is now gone. And that means that the control circuit is disconnected from the flagellum propeller and means the bacterium is effectively immobilized. So we set up an evolution experiment to effectively use this situation as a challenge for evolution. So if you put this immobilized bacterium into an agar plate, that's what that dish is, and you can see the bacterium growing in the middle as that sort of yellowish dot. The bacteria will grow, they will eat the nutrients that they have available, but then they'll get stuck because they can't switch on the flagellum using that FleaQ switch. Of course, there's lots of food around the edge of the plate, but the bacteria will start to starve because they can't go and get it. So that puts an evolutionary pressure on the bacteria to evolve a solution to the problem of missing this gene. And they do exactly that. After 2 to 4 days, the bacteria begin to swim again. That's what that cream-coloured disc is in the middle of the agar plate. And then after 6 to 8 days, they evolve to swim faster as well. So that's what those semicircle shapes at the top and on the right coming off of that disc are. And that shows the bacteria have evolved to swim quicker. Now, this is happening really quickly. 2 to 4 days is about 20 to 30 bacterial generations approximately. So we're talking about a very short time frame evolutionarily, and they're evolving a solution to literally just missing a gene. So this is really quite incredible. When we take a look at the cells under the microscope, we see that before the evolution experiment, the bacteria are just sort of floating around, they're bobbing about in the liquid, but they're not swimming. And I hope you can see that after the evolution experiment, there's a lot more activity there. Cells are swimming around, doing little spiral patterns, and there's lots of activity. So they've evolved the ability to swim again by fixing that gene that we removed. So how is this possible? Evolution is rescuing swimming by hot wiring another control switch gene. This control switch is called NTRC, and it doesn't normally control the production of the flagellum propeller. Instead, it normally controls nitrogen metabolism. So that's getting hold of nitrogen as a nutrient that the cell needs. But now it's being hot-wired to switch on the flagellum. So that's a pretty amazing feat. But this does come at a cost. So whilst it does help the bacterium to swim, by not having the control switch for that aspect of getting hold of nitrogen, the bacteria become a bit sickly. They can't get hold of that nutrient quite so well. So there is a trade-off to doing this. So how is it possible to just hotwire a control switch like this? So it's because the switches are actually similar to one another. They're related. So you can see that these two switches are both little sort of flip switches. If evolution wanted to use another sort of switch like this dimmer switch, it would need to do a lot more mutation in terms of changing that switch into a flip switch. Whereas to change the green flip switch into the red flip switch, not a lot of mutation has to happen. And evolution will always take the easy option. And this is reflected in the protein structures. So these sort of two squiggly shapes below the two switches are what proteins actually look like. And you can see that those two are basically the same shape. So there's not a lot of change that needs to happen for NTRC to be hotwired to do the function that FleaQ does normally. But interestingly, NTRC, the green switch, is not the only similar switch to FleaQ. There's actually a whole family of switches, which are all similar in structure. So the question comes up, why didn't evolution use any of these switches, which would work or could be used to rescue the flagellum and rescue that switch? Why did it use NTRC? So are there any other hot wiring options available? How could we work out why evolution used that gene over another? Well, a good way to do it is to use genetic engineering again and essentially create a situation where evolution is forced to take a different option. And we can do that quite easily by removing this gene as well. So by removing both of these switches, evolution now has to take an alternative if it wants to regain the ability to swim. And we see that this can still happen. So you've got the at the bottom there, you've got the bacteria growing, and then coming off at the top, you've got an evolved swimming bacterium. So it's gained the ability to swim again, but it does this after 2 weeks. So it's taking a lot longer for the evolution of the swimming to occur now that I've gotten rid of NTRC. And what's happening is another switch is being repurposed. It's not got a nice name. It's called PFLU1132. I'll just refer to it as the blue switch for simplicity. But the blue switch is now getting hotwired instead, and it's allowing the flagellum to be switched on. So this is really quite amazing. We've now seen 2 switches that do not normally control the production of the flagellum propeller are now being hotwired to bring its function back online. But when the blue switch is used, it doesn't give a very strong swimming ability. So this is what the blue switch mutants look like when they're swimming in an agar plate. You've kind of got that smudgy sort of haziness, which is where the bacteria are. And then there's all these sort of speckles. You could call it a sort of firework appearance. And that's because the bacteria, or is likely because the bacteria are not swimming very well. So when we measure how fast they don't move as well as the NTRC hotwiring bacteria. So what this shows is immediately we can work out why the green switch, the NTRC switch, is used preferentially by evolution to the blue switch. It's because the blue switch takes a lot longer to evolve, and it also provides a much poorer motility sort of swimming ability than the green switch. So what can we learn from these evolution experiments? They're important for understanding the evolution of those control circuits. So remember I mentioned that these switches exist within a control circuit, a bit like an electrical circuit, which determines which genes are switched on and off. And we found that the control switches within that network can be hotwired and repurposed so that they can control genes that they don't normally determine the function of. And they can even replace the function of lost members of this switch sort of set and family. And this is quite important because these control circuits determine how the information in an organism's genes are turned into the actual organism itself in terms of its behaviors and its structures. So understanding the evolution of these control circuits allows us to understand essentially how does an organism turn its genes into its structures and biology. But this is also relevant to understanding how organisms adapt to challenges. So we've seen that these regulators and switches can be repurposed like this, which is quite an amazing evolutionary feat. In our study system, we look at switches that control genes for swimming, nitrogen metabolism, and maybe environmental survival as well. But this information could easily apply to other switches, for example, ones that control antibiotic resistance, toxin production, or survival in an infection. So there's information to be learned that could be medically relevant in the future. Of course, many of these problems are effectively evolutionary problems. So antibiotic resistance, for example, is where bacteria are evolving to evade and become immune to the antibiotics that we use as treatments for people's infections. So understanding how a control circuit might evolve and adapt to, say, switch on antibiotic resistance genes if we're trying to turn them off is quite relevant and quite important to know. But these findings also apply to all living things, not just to bacteria. So these control circuits exist within all living organisms. For example, in animals, they can determine the body plan. So that's essentially where the legs or the head or the body of an organism goes. So, for example, with fruit flies, you've got this set of genes that form a control circuit, and that control circuit, along with the timing of when switches are turned on or off, determines the shape and structure of an organism. And when mutations happen in these control circuits and switches, we can find some pretty drastic things occurring from very small changes. For example, a small change in the timing of when a certain one of these switches is turned on can lead to crazy things like this happening. So this is a fruit fly's head. On the left, we've got the normal head of a fruit fly. On the right, there are legs coming out of where the antennae should be. So this fly literally has legs coming out of its face. And this is because of a small set of mutations in the control circuit that determines where body parts go. So we can see that very small changes and mutations within control circuits can lead to quite drastic alterations to an organism's behavior, structure, and biology. So thank you very much for listening. I hope I've convinced you that we can use molecular biology to understand the fundamentals of evolution and gain an insight into how organisms might adapt to challenges that they face in their environment. I'd like to thank my supervisors, Tiffany and Lawrence, the members of the Taylor Lab, Louise and James, Pint of Science organisers for allowing me to speak, and of course the Royal Society for that all-important funding. And I look forward to answering any questions you guys might have. Thank you.
Thank you, Matt, and thank you for the wonderful talk. That was absolutely brilliant. If Lewis, our lovely producer, would kindly like to also bring Martin onto the screen, if that's all right. Hello again, Martin. So, time of the talk where we open the floor for some questions. So, One of the questions we have will be coming in from Tessa Flack, and that is, what processing power is needed to render the pig video? It looks quite computationally intensive.
Yeah, I mean it can be, but I think what they've done on YouTube is that they'll actually adjust the resolution of it to fit. So even if you've got a fairly low powered computer, you can or phone, you'll at least get the sensation of it. It'll move around. I mean, I rendered it as high as possibly could. It's in order to get a kind of 4K video effect. Well, actually HD video, you end up having to render at 8K resolution because you're actually only seeing a small section of the whole render, so it can be a bit. I mean, I must admit, because I do computer graphics, I have got a graphics card on my machine, but it still does work. You still do get an effect with the lower power. It just might be a little more pixelated, but it's not fully 3D. It's actually it's actually a video that wraps around, so it's not too bad.
Interesting, nice. So I have one more question before I move to Matt, and that's from Fostek. And do you think the pigs wonder they're being simulated?
Well, I do. I don't know. They seem to be fairly oblivious of what's going on. And the pigs, the farmer said, "Did we want our markers back when we'd finished?" And he said, "We said no, that's all right." And the pig just went back quite oblivious with the marker still on it. So I'm not sure they're really aware of anything. But anyway, thanks.
I'll just like to ask one more quick one, because there's another nice one that's coming from Gemma McCarthy. And that question is there any particular animal you'd like to motion capture in the future, or like?
I don't know. I quite like to do an elephant. I mean, that seems quite a challenge, but I mean, I know people have taken some really challenging things. I know I've heard quite recently someone motion captured a butterfly, and that seems quite extraordinary. I mean, you had very high resolution cameras and they had lots of nets and you had put very small markers on. But yeah, no, that sounds like quite interesting. Maybe a spider would be good. I'd like to sort of, because then of course you motion capture it, then you could make it like 50 feet tall. So when you're in the VR, it would look quite scary. But yeah, there's all sorts of challenges out there. Thanks.
Nice. So moving on to Matt's questions, I have a question from Robin Williams. So Matt, where do you see your research going in the near future?
Oh, cool. Great question. So yeah, so the plan is to essentially work out precisely what's going on when these switches are being hotwired. So we know that the hotwiring is happening because we can see the genes being switched on, and of course the bacteria start swimming around. We don't know exactly what the mutations are doing, so working that out is quite important. I'm going to use a technique called RNA sequencing, which is a bit like DNA sequencing, but it essentially determines which genes are being turned on or off, rather than which genes are present in the bacterium. And then going from there, we've got lots of sort of interesting avenues. Removing the blue switch, so the 1132 switch, that might be quite interesting, because if we get rid of that, then what's evolution going to do? Will it just repurpose another one? How far can we take it? It's quite an interesting sort of avenue, but also trying to work out why evolution takes certain paths over another, and whether we can override the hierarchy as well. So if we were to find a way to sort of make evolution choose a different path in a different situation, that might be quite sort of interesting avenue of sort of further research as well.
Interesting, nice. That's quite good. Look forward to seeing some papers coming out of you then.
Yeah, hopefully.
So another question, and that is from Liam Stubbington. I hope I pronounced that right. And that's, can you change the genetics of your gut microbes by eating specific foods?
I'm not sure you can change the genetics, but you can change the composition by eating certain foods. So pro and prebiotics are quite common. You can find them in the supermarket and stuff, things like Yakult and Actimel, and that's drinking live cultures of bacteria and they're supposed to be beneficial bacteria. But you can also eat certain foods that should promote the growth of beneficial bacteria. So I think things like bananas and onions are supposed to promote the sort of growth of certain bacteria, which is supposed to be helpful. But as for changing their genetics, that would require some pretty fancy technology to do that.
Interesting. one more from Parkin13. And that is, do you think the bacteria would choose a different switch if they were in a poor nitrogen environment?
That's a great question. So that was something we investigated. So if you put these bacteria that have repurposed that nitrogen regulator into a nitrogen poor or limited environment, they are quite sickly. They are struggling in that environment. So it was interesting to see, well, will they use that option in the nitrogen poor environment? And they actually do. They still use it, even though it causes so much damage. But it seems like it is just the easiest option for evolution to go for. It lets them swim, and they can mutate it quite quickly and repeatably. So even though it gets a sort of damaging effect of not controlling its nitrogen uptake so well, it still goes for that option anyway, just because it really wants to swim. Because essentially in our evolution experiment, it's sort of swim or die, really. If they don't swim out from the center of the plate, they're going to starve. So they'll take that option every time.
Interesting. I just have one more last question. I'll pose it back to Martin because he mentioned it the other day in his email. Martin, you mentioned you did some work on the Harry Potter films. What exactly did you do?
Oh, I was mostly on creatures. So there's the hippogriff, did the muscle system, and the tail system on the back of the hippogriff. Yeah, I mean, I got caught actually out in the field behind my house, crouched down videoing the backside of someone's horse. They came out and wondered what I was doing, because that was a bit tricky. And I've also done, there was a whip scorpion spider in a lesson where it sort of crawled down your face. So I modelled the spider and did the rig of the whips and stuff like that. So things like that. And there was this carriage that goes down into Gringotts, all the suspension and stuff like that. So just various. Mostly I was doing rigging and creatures. So that's
fantastic, especially to have had you worked on such a ginormous franchise it is for Harry Potter. That's quite remarkable to see.
Apparently, I have my own wand drawer at the Harry Potter world.
Well, I'd like to thank you both. You've both been absolutely amazing. I'd also like to thank our audience for all the wonderful comments. Thank you again, Matt Shepard and Martin Parsons from the University of Bath. If you want any more insightful talks about any latest developments in science or the latest research that's going on, please visit pintofscience.co.uk to see the schedule of talks over the next couple of days and take part in Big Science Quiz. Tomorrow we've got talks from Manchester, Leeds and Greenwich. But until then, it's goodbye from me and our team here of speakers and the Pint of Science technical team. Thank you. This is University of Bath, and enjoy the rest of your night, everyone.
My name is Adam. I will be your quiz master for this evening. I certainly hope you're going to enjoy yourself. Here we go.